So now we'll move on to look at the general structure of viruses. Viruses are considered to be ultramicroscopic. That means in general they're less than 0.2 microns or micrometers. Most are so small they can only be seen with an electron microscope. Just to give us an example of the scale, if we were to consider a bacterial virus that infects a bacterial cell, you could get about 200 of them inside your average bacterial cell. Or a human virus, say poliovirus. We could fit about 50 million polioviruses inside the average human cell, so they're really, really small relative to the host cell size. Now, obviously, there's a huge variation in size. For example, parvoviruses, which are DNA viruses that cause respiratory infections in humans, are around 20 nanometers in diameter, whereas some of the larger ones, like mimiviruses, are up to 450 nanometers in length. This figure below gives you a good orientation to the scope and the scale that we're dealing with here. The blue area is a yeast cell. Inside the yeast cell, we can see that there's a couple of bacterial cells, like a staphylococcus here and an E. coli over here. So those are much smaller than the yeast cells, which are protozoans. But even smaller than that, we'll see our viruses. The first one here is of a mimivirus. Mimiviruses got their name because they're mimicking microbes, so they mimic other microbes. This is the HIV virus, so it's still fairly large. But as we get down to our smallest viruses, as you can see here, yellow fever is a much tinier virus. They put number 10 in here, which is just a simple hemoglobin molecule, to give us some perspective of the relativity of size of macromolecules and viruses. So essentially, the smallest viruses are not much bigger than a single macromolecule, whereas the largest viruses are fairly close in size to some bacterial cells. Viewing viruses brings up a whole nother set of special circumstances. They're so small that they cannot be seen through a light microscope, so they require special stains as well as an electron microscope. We looked at some of these techniques earlier in the semester, but negative staining can be used to outline the shape. Positive staining can show some of the internal details. And then shadow casting technique is another technique that's used to view viruses. As we begin to consider viral components, you'll notice that because viruses don't have the capacity to live without the machinery of other cells, they don't need any of that extra stuff inside. So relative to the other microbes that we've observed so far, viruses will seem fairly simple. They're composed of simply capsids, nucleic acids, and envelopes. The molecular structure is composed of regular repeating subunits. This gives rise to the crystalline appearance that you can see here in this figure. This is magnified 150,000 times, and you can see that there are hundreds of individual viruses in this image. This is a purified poliovirus crystal image. Viruses contain only those parts that are needed to invade and control a host cell. Those components are an external covering and then a core of genetic material. The basic viral structure again consists of an external coating and then a nucleic acid core. This external coating is a protein capsid and sometimes an envelope over that. The envelope is found in 13 of the 20 families of viruses that infect animals. Some viruses have the envelope, some don't. If there is no envelope, it's called a naked virus, and if there is one, it's called an enveloped virus. As far as the core, it's a nucleic acid core, so it's either DNA or RNA. Again, that DNA can be single-stranded, double-stranded, and also the RNA could be single or double-stranded. The capsid and the nucleic acids together are termed a nucleocapsid. This is just a simple virus with no envelope. A fully formed virus that's able to establish an infection in a host cell is termed a viron. You can see in these images here an example of a naked nucleocapsid virus. And on the right, we see an enveloped virus. Let's take a closer look at the protective outer shell, or the viral capsid. It's constructed from identical subunits called capsomers. They're made up of protein molecules. There are two different arrangements of capsomers. There's the helical arrangement and the icosahedral arrangement. First, let's look at the helical arrangement. You can see this helical arrangement in the top figure here. Capsomers assemble into hollow discs, so each blue subunit in here is a capsomer. And they've assembled in a structure such to form a disc that's hollow in the center. The nucleic acid is then inserted into the center of the disc. And then the elongation process for the nucleocapsid results from addition of capsomers. As the capsomers are added, the nucleocapsid could grow from either end or both ends. The nucleic acid is then wound within the lengthening helix. You can see an example here of the tobacco mosaic virus. It's a naked helical virus. Below that, we see the influenza virus. The influenza virus is an enveloped helical virus. We're looking right down the barrel of it here. The icosahedral form is a little bit more complex. It is a three-dimensional 20-sided figure with 12 evenly spaced corners. You can see this three-dimensional model in full form here. Let's break it down just a little bit though. Up at the top, we can see the capsomers. Here, there are 21 different capsomers arranged in a triangular form. At each point, this face comes to meet with other faces. As they meet, there's somewhat of a unique structure that arises. Now let's put this face in place and see how the vertices and facets or faces come together. And again, we can look at our three-dimensional model and see how that manifests itself. This model represents magnification of about 640,000 times. Below, you can see a negative stain of this same structure. It highlights the various textures on the surface. Now, there are some forms of viruses that don't fit into either helical or icosahedral. So another category was created to fit those into, which is called complex. Complex capsids are mostly found in bacteriophages. Bacteriophages are viruses that infect bacteria. This is a common complex form of a bacteriophage. You'll notice the shape is not symmetrical, and there are multiple types of proteins that come together to form this complex structure. It also contains tail fibers through which the phage is able to inject its DNA into the host. And the capsid head is where the nucleic acids are held. I think these look much like little moon landers. And they do that. They're little moon landers. They come along, they land on the cell, they squat down and puncture with the tail pins and inject the nucleic acids that are in the capsid head. Let's have a quick concept check. What is the protein shell that surrounds a viral genome called? Is it an envelope, a capsid, a core, or a cortex? Well, I think this one's pretty straightforward. We're only dealing with envelope and capsid, and the capsid is the only one that's protein. Now let's look at the viral envelope. Envelope viruses are normally animal viruses. And they get their membrane by taking a bit of the host cell membrane and forming the envelope. They'll either bud out of the cell membrane itself, or some of them will leave the cell wrapped in a piece of nuclear membrane, or wrapped in a piece of endoplasmic reticulum. Either way, they're going through the endomembrane transport system because the membrane of the nucleus and the ER is continuous. And so they'll wrap themselves in essentially a vesicle that'll then move through the cell and out to the cell membrane. In the envelope, some or all of the regular membrane proteins are going to be replaced with viral proteins, and these proteins are often involved in binding the envelope to the capsid. Some other proteins are associated with carbohydrates, or they're glycoproteins, and they remain exposed through the cell envelope as spikes, and these spikes are essential proteins for attachment to their host cells. The outer layer, considering capsids or envelopes, are essential in viral function as they act as a barrier that protects the virus. For example, enteric viruses or intestinal viruses like polio and hepatitis A are resistant to the acid and protein digestive enzymes that we find in the gastrointestinal tract. Also, the barrier layer is involved in introducing the viral DNA or RNA into a suitable host cell. They help the virus bind to the cell surface using their very specific special binding proteins. And they also assist in penetration of the viral nucleic acid. We most recently looked at the tail fiber proteins in the complex capsid. These proteins found in the capsid in the envelope also stimulate the immune system to produce antibodies that protect us or other host cells against future infections from that same virus. Let's have a quick concept check. What is the lipid membrane that surrounds some viruses called? Is it a capsid, an envelope, an outer membrane, or a capsule? I think this one is as simple as the last one. We're really limited to capsid or envelope, and the envelope is the lipid membrane portion. So on we go. Let's take a look now at the nucleic acids at the core of the virus. The sum total of all the genetic information that's carried by any organism is called its genome. This goes for viruses also. Now, a virus doesn't need as many genes as any other organism, because all it needs to do is instruct its host cell to make viral proteins. So it doesn't need to make any proteins for itself in its normal metabolic functioning because it hijacks the host. Of course, the number is very varied. For example, hepatitis C has only about 4 genes, as compared with the herpes virus that has hundreds. But this isn't many at all when you consider bacterial cells like E. coli have upwards of 4,000 genes in their genome. This is because the virus only needs genes necessary to invade the host cells and redirect their activities. Some viruses are exceptions to the typical DNA and RNA formations. Some can contain single-stranded DNA, like the parvoviruses, and others could contain double-stranded RNA, which is unusual, like reoviruses. This figure shows some medically relevant DNA viruses of eukaryotes. First, we divide them into enveloped versus non-enveloped strains, and then we characterize them by their genome type. For example, a double-stranded genome, a double-stranded genome in non-enveloped, or a single-stranded genome. When we consider RNA viruses, most of them are single-stranded. As you'll probably recall from general biology, information usually flows from DNA to RNA to protein. However, the discoveries in viruses change things a little bit. Some viruses are packaged with RNA, and the RNA will work in a variety of ways as we see. There's positive sense RNA. This is a virus whose genome is ready for immediate translation for proteins, so the RNA codes directly for all the machinery that the virus needs in order to take over the cell and break out. Negative sense RNA means that the virus's genome has to be converted into the proper form to be made into proteins. So we might need to transcribe a positive sense piece of RNA from the negative sense RNA. Sometimes we'll also see that the individual genes exist on separate pieces of RNA. These are considered segmented RNA viruses. Let's take a quick look at some medically relevant RNA viruses. Again, we first divide by enveloped and non-enveloped. And then the type of genomes, single-stranded genomes or single-stranded genomes that encode reverse transcriptase. We'll deal with that in a little while. Or we could be dealing with double-stranded genomes versus single-stranded genomes, or even segmented genomes. These are ways that we categorize our viruses so we can place them in manageable groups. Here's an example of the diversity of viral genomes. Remember, they can only have one kind of nucleic acid, either RNA or DNA, but not both. So that makes them unlike cellular organisms also. You don't need to commit these to memory, but DS means double-stranded and SS means single-stranded. And you have the denotation of positive or negative, whether they're positive strand, meaning they're totally ready for translation, or they're negative, which means we need to make a positive copy first. There are some other things that we find inside viruses. As much as they don't have enzymes for metabolic processes, occasionally there's a need for them to produce an enzyme that helps them take over the functions of the host cell. So again, there are no genes for metabolic enzymes, but occasionally we'll see a functional enzyme. For example, we might see polymerases that might allow the virus to copy its own DNA and insert it into the host's genome. Reverse transcriptase is a perfect example. We see this in the HIV virus. HIV virus is an RNA virus, and it reverse transcribes DNA from RNA. So it inserts its own genes into the host's genome by reverse transcribing its RNA gene and making a DNA copy that's now encoded into the host's DNA. Here are two examples of enzymes that are found in the envelope, hemagglutinin and neuraminidase. We'll cover those later. While we're here, it's worth taking a moment to look at how viruses are officially classified and named. In an informal and general way, we've already begun to classify viruses as either animal, plant, or bacterial viruses. That is, they infect those specific organisms. They could be enveloped or naked, they could be DNA or RNA, then helical or icosahedral. These categories are really useful in organization and description, but of course there is a much more standardized, more complicated method of nomenclature. This method is based on three main criteria. There's the virus structure, its chemical composition, and the similarities in genetic makeup. The International Committee on Taxonomy of Viruses in 2005 defined 3 orders, 73 families, and 287 genera of viruses, as compared to in 2000 when we only had 1 order. So this field is expanding at a very fast rate. You'll notice that the family names all end with viridae, and the genre names all end with virus. For example, Filoviridae Ebola virus. Some virologists use a species naming system, but it's not an official designation because things in viruses change so fast. And being as they're not living, how can they possibly speciate? The table below gives some examples of the 3 orders of viruses. You don't need to commit them to memory. We'll be covering several different viruses throughout the course of the semester that we should find really interesting. So for now, we'll call this the end of our section on viral components. And get ready to move on to look at modes of viral multiplication.